Welcome to the final segment of Chapter 17. In this segment, we're going to talk about different types of mutations and how they affect the structure and the function of proteins. Let's go ahead and do a little quick review of the genetic code. So if you remember the codon table, the codon table lists all the potential codons and the amino acid in which they code. Remember, there are multiple codons per amino acid. However, each codon only codes for one amino acid, so it's non-ambiguous. Now, these are going to become important when we are trying to talk about what happens to proteins when the codons or the nucleotides within the codons change due to mutations. Now, mutations are changes in the genetic material, really in the DNA, of a cell or a virus. Mutations can occur with one or more than one nucleotide, and of course is going to affect the protein structure and the function of the protein if changes downstream occur as well. Now, point mutations are changes in just one base pair of a gene. The change of a single nucleotide in the template strand of DNA, remember the template strand actually codes for the protein, this can lead to the production of a protein that is not normal. And we'll talk about how in just a moment. Now, point mutations can fall into one of two categories. Now, one of them is just a single nucleotide pair substitution. So that's basically let's say an adenine is changed to a cytosine, which will result in a complementary base pair being a guanine rather than a thymine. Now, it could also occur in nucleotide insertions where a base is inserted where it should not be. So there's an extra base in a gene. And then, of course, alternatively, a nucleotide can be deleted so that a gene is missing a nucleotide pair. Now let's look at an example of a substitution and what can happen downstream, and then we'll see some examples of each one. So this is a common example that's often used. Remember the mutation in hemoglobin. So remember, hemoglobin is that protein that's found in blood and it carries oxygen. Now, we have a normal structure for hemoglobin, which has its normal sequence, which would result in the normal protein. But then you have individuals who have sickle cell disease. And sickle cell is the result of at least one mutation, and this is the key mutation that occurs. So we're going to compare this to the wild-type hemoglobin. So here we can see the wild type has this codon would be CTT-GAA. Now this is in the DNA, so when the template strand is transcribed, and the codon is actually GAA. So the template strand here would be the 3' to 5' strand in this wild type hemoglobin. And this codon codes for glutamate. Okay, so this is the normal amino acid at that position. Now, with those who have sickle cell, what is typically the issue is that there was a mutation in this particular codon where instead of CTT, it's CAT. So an adenine was incorporated into the DNA where there should be a thymine. So this results in when the template strand is being transcribed, instead of GAA, it's GUA, and GUA actually codes for a different amino acid, so that in those individuals with sickle cell, they have a valine at this position rather than a glutamate. Now this is kind of important, and I'm going to kind of expand on some things that you learned early in the semester. So glutamate is a negatively charged amino acid, so it's charged. Valine is a hydrophobic amino acid. Okay, so they have different chemical properties, and this particular amino acid position happens to be on the surface of the protein, hemoglobin. Now, glutamate on the surface isn't so bad. Remember, it's negatively charged, so it can hydrogen bond with water and other like things, so it's hydrophilic. The valine, on the other hand, is hydrophobic, and this being on the surface and interacting with water does not go very well. So remember that oil and water analogy. So what ultimately happens is, and I think I've discussed this before, is that since the valine is there, it's hydrophobic, it's on the surface, the hemoglobin molecules like to form these very long fibrils. And remember, this is the beta hemoglobin subunit. So it's only one of the subunits. However, again, it is on the surface and it forms these long fibrils inside the blood cells, creating that sickle shape. And of course, that results in the downstream phenotypic effects of sickle cell disease. Now let's talk about some other types of mutations. Now, we just talked about a single pair substitution. Okay, so this is where one base replaces another one, and a number of results can happen, and there's special terms for them. One of them is a silent mutation. The other one is called a missense mutation, and the final one is called a nonsense mutation. Now, these are referring to what happens downstream in the protein. Now, what I just showed you with sickle cell, this is an example. That is an example of a missense mutation. This happens when a nucleotide pair is changed at the DNA level and then results in a change in the amino acid. Now, if the amino acid is not changed, it's called a silent mutation. It's silent because you do not notice an effect, right? So it's there, it's a mutation that's there. However, it has no effect on the function of the protein and, of course, no effect on the individual. A nonsense mutation is where an amino acid is changed, however, it's changed to a stop codon. So remember, during the process of translation, once a stop codon is reached by the ribosome, it stops translation. So it stops making that protein. So if that stop codon is too early, then the protein is going to be shortened, and of course, its function is going to be severely affected. Now, let's look at some examples of a particular gene and see the different effects of all these different types of mutations. Now, we have a template strand of DNA here, and then we have it transcribed into messenger RNA. So the template strand is that top one, and then the messenger RNA, of course, is going to carry the codons. And you can see this messenger RNA codes for this protein right here, methionine, lysine, phenylalanine, and glycine, and then here's the stop codon. So let's say this is the wild type gene or protein, right? So this is the normal gene or protein. Now, let's say that an adenine is replaced or replaces a guanine. Okay, so in this position up here, we have the guanine. Instead of guanine, we have adenine now. So what happens is this codon is actually changed, right? So we have right here C or GGC. Now it's GGU. However, both of these codons code for the same amino acid. So this would be an example, of course, of a silent mutation. Now, a missense mutation, let's see this example right here. So instead of a cytosine, a thymine was incorporated. Okay, so we have the cytosine up here, we have the thymine down here. So this codon would normally be still, again, a GGC. However, when this thymine is incorporated instead of the cytosine, the codon changes to AGC. Now, this is the normal one. Glycine would be the amino acid that's incorporated to that position under normal conditions. But after this single nucleotide pair substitution, instead of glycine, we have serine. Okay, so that changes the amino acid at the protein level. This is an example of a missense mutation. Now, let's look at a nonsense mutation. So here, an adenine was incorporated instead of the thymine, which is up here. So normally in the protein, a thymine would be here. This would result in AAG being the codon, and that codes for lysine. However, incorporating an adenine instead of that results in a stop codon. So now the translation is going to be prematurely stopped, and the protein is going to be truncated, meaning it's going to be shortened. And now, of course, it is not going to have the same function, if any function at all. That's an example of a nonsense mutation. Now let's talk about what insertions and deletions can do. So as you remember, I already discussed what an insertion and a deletion is. So an insertion is an addition of nucleotide pairs, and deletions are basically losses of nucleotide pairs. These types of mutations have the most disastrous effect on proteins. Insertions and deletions of nucleotides can alter what's called the reading frame. So the reading frame is basically the order in which the codons are read during translation. And this is going to produce, if there's an insertion or deletion, it will produce what's called a frame shift mutation. So basically it shifts the frame either left or right. So it's going to shift the frame to the right if it's an insertion, because remember, bases are added. And then if bases are deleted, it's going to shift the frame to the left. So let's see this effect. So here is that same wild-type piece of DNA with the same protein. Now, let's say an adenine was inserted here. Okay, so this is an extreme example. Now, inserting an adenine here can result in in this case, it's resulting in a premature stop codon. So this is a frame shift mutation and also a nonsense mutation. So you can see nonsense. But its original result is due to a frame shift. Now, let's look at another one when we delete an amino or a base. Okay, so again, same DNA sequence and protein, and over here we have an adenine being deleted. Okay, so this results in a frame shift to the left, and instead of having UUU coding for phenylalanine, we have UUG, which codes for leucine. Now, it's important to remember that when there's insertions or deletions, wherever that insertion or deletion is, if it changes an amino acid just to another amino acid, all of the other amino acids after that can potentially be changed. So in this case, since we're shifting to the left, instead of glycine being the next amino acid, that codon shifts over and it becomes alanine. Okay, so you can see the severe effects on proteins that any mutation can have. However, insertions and deletions have the greatest effect on the function and structure of the protein. Now, you can do this as well. So let's look at this example we have where 3 bases have been deleted. However, these 3 bases happen to be exactly a codon from the normal protein. So basically, in this case, just an amino acid is deleted. Now, this you would consider that it potentially could have a severe effect on the protein. However, it's less than an insertion or deletion of only 1 or 2 bases, especially if it's just a codon, because you're just removing 1 amino acid. Now, it depends on what the purpose of that amino acid is. If it has a function or a direct function in, let's say, enzymatic activity, then yes, it's going to have a severe effect on the function of the protein. But in most cases, it could be somewhere else, and it will not be as bad. Now, mutagens are chemicals or some sort of physical effect that can cause mutations in DNA. So spontaneous mutations are just ones that are basically mistakes during DNA replication or during recombination and or repair. Okay, so that's basically mutations that can occur during normal processes within the cell. But mutagens are actual agents that can cause mutations. So mutagens can be physical or they can be chemical. A main example of a physical one would be UV light. UV light is very dangerous. There's actually UV that's used as a disinfectant or to sterilize surfaces such as those surfaces that might be found in the hospital. Here's some other examples of some mutagens. So some of them would be considered in different classes, like carcinogens, clastogens, teratogens, and then there's just ones that are more non-specific, and their effects, right? So carcinogens will result in the formation of tumors. And these are some examples right here. So X-ray radiation, we have UV rays, which can be considered a clastogen. X-rays can also be a carcinogen or a teratogen. And so the clastogens are going to cause breaks in the chromosomes, deletions, and rearrangements in the chromosomes. So you can think about what you've learned already and kind of imagine the disastrous effects of these types of mutagens. Teratogens cause congenital malformations, so this is going to affect the unborn baby at some point. So it'll affect your gametes and cause mutations in the gametes. Then the nonspecific mutagens can cause all types of damage to genetic material. And again, some of the examples overlap. For instance, X-rays can be considered carcinogens, teratogens, and just nonspecific mutagens. Now, in summary, I talked about mutations, and most things are point mutations. So that's a single base pair substitution, change in bases, or insertion and deletion of single bases. However, it's important to remember that multiple bases can be changed, and also multiple bases can be inserted or deleted. Now, their effects on proteins can result in one or more things, right? So we have a missense mutation, which, remember, changes the amino acid, which changes the structure of the protein and probably function. A nonsense mutation introduces a stop codon where it should not be. A silent mutation has no effect on the protein. And a frame shift mutation can result in significant effects or less significant effects, as we saw in the examples. And then, of course, I just spoke of the mutagens being of two different major classes. They could be physical and or chemical.